Okay, everybody, welcome back to seminar. Before we begin, I just want to make one announcement that we've heard from some of the people who attend the seminar via Zoom that during the question and answer, they're having a little bit of difficulty hearing some of the questions. So if you have a question today, just be sure to speak up so that the viewers on Zoom can hear as well. So our speaker today is Hannah Smith. And she's going to be presenting on some of the diet-related effects on muscle health. Hannah completed her Bachelor of Science in Civil Engineering and recently defended her master's thesis in Biomedical Engineering, both at the University of Calgary. She's currently helping out with a few projects going on in the Herzog lab and would like to eventually go into medicine. An interesting fact about Hannah is that she's an adoption coordinator at a local dog rescue and loves to hike and spend time in the mountains. So we're looking forward to your talk, Hannah. Thanks.
Thank you. Can everybody hear me well? Okay, awesome. Thank you everybody for coming to my HPL seminar. The title of my talk is Examining the Effects of Diet on Muscle Health in a Female Diet-Induced Obesity Rat Model. So since 1975, the number of people with obesity has nearly tripled worldwide, with 1.9 billion adults and 379 million children considered either overweight or obese. And obesity is defined by an excessive fat accumulation that has been demonstrated to put people at an increased risk for many different health conditions. The combination of the availability of high energy foods and the decreased need to move in today's society has caused individuals to store excess nutrients as fat in their bodies. And so this places significant stress on the body's metabolic pathways and can lead to numerous non-communicable diseases stemming from metabolic dysfunction. Metabolic syndrome is associated with obesity and is defined by a cluster of conditions that include visceral obesity, hypertension, dyslipidemia, and elevated fasting glucose. Obesity and metabolic syndrome put an individual at an increased risk of diabetes, cardiovascular diseases, MSK disorders, and certain types of cancers. And it is particularly interesting to look at MSK disorders in the context of obesity, as damage to MSK tissues can lead to the worsening of obesity and metabolic syndrome due to the increases in sedentary behavior. And so recently there's been evidence that the rise in metabolic diseases is associated with the increased risk of certain MSK diseases, such as sarcopenic obesity. Sarcopenic obesity is defined by a loss of muscle mass and strength coupled with an excess fat mass, and has been associated with an increased risk of comorbidities and mortality and a reduced quality of life. An important feature of sarcopenic obesity is fatty infiltrations in the muscle tissue called myostatosis. And this relative loss of muscle mass to fat gain can exacerbate certain symptoms of metabolic syndrome and can contribute to insulin resistance and the onset of diabetes, as skeletal muscle is the largest organ in the body responsible for glucose regulation. Fat is also an active endocrine organ that releases inflammatory cytokines, and intramuscular fat deposits within the muscle can release these inflammatory cytokines and create a chronic inflammatory environment within the muscle. And muscle is a very vulnerable tissue in an inflammatory environment, such as the one caused by obesity, as it is highly plastic and undergoes regular remodeling triggered by inflammatory signaling. And so this environment created by the local cytokines creates conditions of chronic injury leading to irregular remodeling and pathophysiological fibrosis that can lead to the worsening of physical function. So our lab many years ago wanted to study the implications of obesity on muscle degeneration. And so we created a preclinical model of diet-induced obesity using male Sprague Dawley rats. At this time, only male animals were considered for the model, as they are typically thought to have a more steady metabolic profile, while females undergoes periodic cycles associated with their childbearing function. And so in this model, it was demonstrated that a high-fat, high-sucrose diet induces obesity and low-level systemic inflammation. And the primarily glycolytic VL or vastus lateralis muscle showed an increase in fat infiltration and fibrosis and local inflammation in as early as 3 days following the start of a high-fat, high-sucrose diet. However, the soleus muscle seemed to be protected against this increase in intramuscular fat infiltration and other changes in the structural integrity of the muscle. And this was hypothesized to be due to the protective effect of the oxidative capacity of the soleus muscle protecting against the fat infiltration. And so it was also found during these studies that a strong relationship existed between body fat accumulation and intramuscular fat infiltration. And this study also confirmed the link between the intramuscular fat infiltration and fibrosis, as strong correlations were found in the VL muscle. So fortunately, obesity can be modified through certain diet interventions, such as oligofructose prebiotic fiber supplements. Oligofructose is a prebiotic fiber that is unable to be digested by humans, but it is fermented by the gut microbiota to produce short-chain fatty acids that stimulate hormones associated with appetite reduction and delayed gastric emptying. And so this has been shown to decrease body fat percentage and improve the metabolic health of both rodents and humans. So other studies have also shown that oligofructose fiber supplements induces a change in the composition of the gut microbiota, and that decreases the permeability to bacterial lipopolysaccharides, or LPS, thus decreasing the systemic inflammation associated with obesity. So to counter the damage associated with obesity, previous studies have successfully fed a prebiotic fiber supplement coinciding with the start of a high-fat, high-sucrose diet to prevent knee joint degeneration in male Sprague Dawley rats. However, there are no studies currently that look at the effect of the prebiotic fiber supplement on intramuscular fat infiltration as a consequence of the high-fat, high-sucrose diet. So these interventions where the fiber is introduced simultaneous to the high-fat, high-sucrose also don't mimic typical human eating behaviors, as a fiber supplement would be more likely to be introduced after the onset of obesity and subsequent musculoskeletal problems. And as I mentioned before, these studies have only been done in male rats, and as female animals can differ in the presentation and mechanism behind many diseases, it is important to understand the impact that sex has on musculoskeletal health. And so that brings me to the objectives of my thesis. Their overall objective was to explore the effects of diet on muscle health in a female diet-induced obesity rat model. And we accomplished this with 2 different studies. So the first study was aimed at determining the sex differences in a diet-induced obesity model. And then the second study was to determine the effect of a 3-week delayed prebiotic fiber supplement in the same diet-induced obesity model. So for the first study, we wanted to develop and validate a diet-induced obesity model in female Sprague Dawley rats and determine if there were sex-related differences in the response to the high-fat, high-sucrose diet with respect to muscle integrity and the development of metabolic syndrome. And so we hypothesized that the female Sprague Dawley rats would respond similarly to the males after a 12-week high-fat, high-sucrose diet intervention. And we predicted that the female animals will develop the same symptoms of metabolic syndrome and have compromised muscle integrity. So to do this, we had 24 males and 20 females, 12-week-old Sprague Dawley rats that were randomized into 2 different experimental groups per sex. And so one group was fed a rodent chow diet, which is consistent of that of a balanced diet. And the other group was fed the high-fat, high-sucrose diet, which mimics a Western type diet. So 39% of the calories come from fat and 43% come from sugar. And these animals remained on their respective diet interventions for 12 weeks. And at 24 weeks of age, they were sacrificed and their tissues were harvested for analysis. At the endpoint of the study, we collected data related to the different markers of metabolic syndrome. We used the DEXA to measure body composition and body weight. Before sacrifice, we collected blood so that we could analyze the blood lipid profile and serum cytokines. And we also performed an oral glucose tolerance test to determine insulin resistance. And then in terms of muscle integrity and inflammation, we harvested both the vastus lateralis and soleus muscle. And we chose these muscles for two reasons. They represent the different muscle types in the animal, the VL being fast twitch and the soleus being slow twitch. And also these muscles have been analyzed in previous studies. And so we wanted to see if we could make comparisons to previous results. And so we performed histology and immunohistochemistry on both of these muscles. We used Oloretta staining to measure intramuscular fat, Picroceros red to measure fibrosis, and then we used CD68 immunohistochemistry staining to give an indication of macrophage infiltration, which measures the local inflammation. In terms of statistical analysis, we use non-parametric Mann-Whitney U test to measure the data pertaining to metabolic syndrome and systemic inflammation. And we used a two-way ANOVA that was performed to see if any significant interactions between diet and sex existed on the histological analysis of muscle integrity. And then finally used a least square linear regression to determine if a relationship existed between body fat and intramuscular fat. And so this is our first set of results. We can see this graph of body fat. We have the both male and female animals. The chow animals are in the gray box, and the female animals are in the white box. And so we can see that between the two diet groups for both males and females, the animals that received the high fat, high sucrose diet had an elevated body fat percentage compared to the animals on the chow diet. Next, looking at dyslipidemia, we measured triglycerides, HDL cholesterol, and total cholesterol. And we did find that both males and females had an increase in the blood triglyceride levels after receiving the high fat, high sucrose diet. However, only the females had an increase in cholesterol after the high fat, high sucrose diet. And then looking at insulin resistance, it was seen that both males and females experienced an increase in insulin resistance. And so based off the markers of metabolic syndrome, increased body fat, markers of dyslipidemia, and insulin resistance, both the males and female animals exhibited all of these symptoms. And so now looking at the results from the VL and soleus morphological changes, for the VL, the two-way ANOVA showed that there was a main effect of diet on intramuscular fat, which we can see in this graph here. We have the animals that receive the high fat, high sucrose diet have a significant increase in intramuscular fat. However, for the soleus, the two-way ANOVA revealed that there was a main effect of diet and then also a diet and sex interaction, indicating that the level of the effect of the high fat, high sucrose diet was dependent on the sex of the animal, which we can see in the bottom graph that only the females seem to be affected by the increased intramuscular fat infiltration. And so the large amount of fat infiltration found in the soleus muscle of the female was the most unexpected result from this study, as it is contrary to previous findings where we found that in the male animals, the soleus was protected against that intramuscular fat infiltration. So a possible explanation may be how males and females store fat differently than each other. It is thought that estrogen mediates fat deposition preferentially away from visceral fat and towards subcutaneous adipose tissue. And so it's possible that this direction away from visceral fat is also directing fat into the muscles. And so directing fat, and then from an evolutionary perspective, because visceral fat contributes more to metabolic dysfunction, not having the fat deposited elsewhere could be protective for reproductive functions as well as caring for their young. So in conclusion of this first study, we were able to develop a model of diet-induced obesity in female Sprague Dawley rats, and we showed that these female rats were susceptible to obesity in the same way as the male rats and also showed the same symptoms of metabolic syndrome. We also found that sex differences do exist in how females store fat in the body. especially in the soleus muscle when exposed to the high-fat, high-sucrose diet. And including females in this research may allow for a better understanding of the relationship between diets, obesity, and MSK degeneration. And so that brings us to our second study. In the study, we wanted to determine whether a 3-week delayed prebiotic fiber diet intervention alleviates or modifies the effect of the diet-induced obesity on muscle integrity and metabolic syndrome in the female Sprague Dawley rats. And so we hypothesized that there's a sort of critical timeline that exists for the fiber diet intervention to be implemented to have a positive effect on skeletal muscle integrity. And so for this study, we had 44 female Sprague Dawley rats that were randomized into 4 different experimental groups. 8 animals were fed a rodent chow diet, 12 were fed the high-fat, high-sucrose diet, 8 were fed the high-fat, high-sucrose diet with a 10% oligofructose fiber supplement, and then 12 were fed the high-fat, high-sucrose diet for 3 weeks, and then after 3 weeks were supplemented with that same 10% oligofructose fiber supplement. And then these animals also remained on their diets for 12 weeks and were sacrificed at 24 weeks of age. And for this study, I used the same methods and data collection mentioned in the previous study, so I won't go through it again in detail, but we took measures for body weight, body composition, blood lipid profile, muscle histology, and immunohistochemistry for both the VL and soleus, and looked at the blood serum cytokines. And then in terms of statistical analysis, we did non-parametric Kruskal-Wallis H test to determine any differences between groups. And so again, we have a group for that looks at the body fat percentage of all the different groups. Each of the groups are in different shades of gray. And it was found that all groups that received the high-fat, high-sucrose diet in some way, whether it was or not supplemented with the fiber, they significantly increased in body fat percentage compared to the chow control group. For the blood lipid profile, we found that only the high-fat group with the high-fat, high-sucrose had increased blood triglycerides, and only the high-fat, high-sucrose group with the delayed fiber intervention had increased cholesterol, which is consistent with aspects of dyslipidemia. So the group that received the fiber supplement at the same time as the high-fat, high-sucrose diet seemed to be protected against this. And so although they all saw an increase in body fat percentage, it seemed that the fiber that was introduced at the same time had an additional protective effect against the dyslipidemia associated with metabolic syndrome and obesity. And then in terms of muscle morphological changes in the VL and soleus, we found that the simultaneous fiber supplement was able to prevent fat infiltration in both the VL and soleus. However, the delayed fiber supplement was only able to prevent fat infiltration in the VL. And then, although the data shows that it was effective at preventing fat infiltration, there are quite a bit of variability in the data in each group. And because Sprague Dawley rats are bred to maximize the genetic diversity in their population, these can cause the animals to have quite a variable response to the diet. And so it has been shown previously that intramuscular fat is tightly linked to body fat percentage. It is possible that a higher sample size would have revealed a significant difference in the intramuscular fat infiltration. And so finally, we have some results here that look at the serum cytokines that were measured in this study. It was found that the inflammatory cytokines were higher in the high-fat, high-sucrose and the high-fat, high-sucrose plus fiber delayed group, indicating that the 3-week delayed prebiotic fiber intervention did not protect as effectively against the systemic inflammation associated with obesity. And so the cytokines that we measured, IL-1 alpha, TNF alpha, IL-12P70, and IL-17A have all been linked to the systemic inflammation associated with metabolic endotoxemia. And so our results suggest that these markers of metabolic endotoxemia were attenuated when the fiber supplementation was introduced at the same time as a high-fat, high-sucrose diet, but not when it was delayed by 3 weeks. And so this suggests that some sort of change happens in the first 3 weeks to the gut permeability during the initial stage of consuming the high-fat, high-sucrose diet that isn't able to be reversed later by a prebiotic fiber diet supplementation. And so in conclusion, our study is the first to our knowledge to use an animal model to consider the effect of a delayed prebiotic fiber diet supplement on metabolic condition and muscle integrity. And we found that the 3-week delayed prebiotic fiber intervention was not able to prevent against the systemic inflammation or intramuscular fat infiltration due to the high-fat, high-sucrose diet in the soleus muscle specifically. However, the delayed intervention was still able to prevent an increase in fat infiltration in the VL muscle. And so this study suggests that prebiotic fiber may be more effective at preventing obesity-related metabolic and muscle dysfunction than reversing the damage at later stages of the disease. So there are a few limitations that need to be discussed for the previous studies. The first being the small sample size of the 2 groups of animals, the chow and the high-fat, high-sucrose plus fiber group included in study 2. And as I mentioned, the outbred nature of the Sprague Jolly rats maximizes the variability in our results. So because of the low sample size, our statistical tests may not have been powered adequately to detect differences in the results. Another limitation is that we did not include a muscle tissue level inflammatory analysis and only looked at the systemic inflammatory profile. And it is more likely that the local muscle inflammation is different than the systemic state. And is probably more important in relation to muscle adaptation. And then lastly, we did not consider any aspects of how the fat infiltration might affect like physical function, such as muscle strength. However, it has been seen and well documented in literature that increases in intramuscular fat and fibrosis are associated with weakened and decreased physical function. And then in the future, it would be useful to for studies to aim to quantify the effects of the female sex hormones and determine if there is a relationship between sex hormones and intramuscular fat allocation. And an ovary-ectomized female rat model could be useful to be utilized to eliminate circulating sex hormones to help determine their role. And then another important direction to consider is to look into those early changes in intestinal permeability, as our results did suggest that the high-fat, high-sucrose diet induces early changes to the permeability of the intestinal lining soon after the animals were exposed to it. And so 2 biomarkers that can be measured in blood serum that are easily to be tested for over time are GLP-2 and LPS. And then finally, in summary, our objectives for this thesis were met by exploring the validity of a diet-induced obesity model in female Sprague Jolly rats, and we found that sex differences do exist in how males and females store intramuscular fat. And our second objective was to explore the simultaneous and 3-week delayed prebiotic fiber supplement, and we found that the delayed fiber supplement was not as effective at preventing intramuscular fat infiltration and systemic inflammation as a simultaneous intervention. And so these findings demonstrate the importance of including female animals in research as they are potential sex-related differences that would not have been elucidated otherwise, and that might provide crucial insights into mechanisms that relate a high-fat, high-sucrose diet to obesity, metabolic syndrome, and muscle degeneration. And then these results also call for further investigation into initial changes in the gut barrier integrity. And with that, I want to thank my supervisor and everybody involved in this project, and I'd be happy to take any questions.
Cool. Thanks a bunch. Do you have any questions? I'll start with the first one. So I just have a question about the fiber type effects that you saw and the sex differences, kind of that interaction. So in
we know in some animals they have like these muscles that are primarily fast or primarily slow. In humans, though, there's more mixed fiber types. So do you think that those results are relevant to humans or like translatable?
From what I've looked at in humans in the literature, it's what's more likely often found is more similar to the female type. It seems that the muscle fiber types or the primary muscle fiber type of the muscles don't really have an effect on intramuscular fat infiltration. In the literature, it shows like with the high fat, high sucrose diet, they've seen an increase in intramuscular fat infiltration in all different types of muscles.
So because they're all kind of mixed, then they all have that.
Yeah.
Okay. Any other questions? Venus?
So I'm wondering why the 12 weeks, like why do you need to have this duration of 12 weeks prevention? Why in the introduction you said even at 3 weeks, so 3 days, you start seeing some changes?
Yeah, so by 12 weeks, obesity has kind of become like a chronic condition, and it's gotten more, I guess, established, and the body's, I guess, has started to like adapt to the changes associated with obesity. So it's just more consistent, whereas at 3 days, there can be a lot of acute responses to the body. So at this point, at 12 weeks, it's been kind of been adapted and consistent. Yes, I know that when this model was established, there was a whole spectrum of lengths of the high fat, high sucrose diet that was used. I know like 3 days. Kelsey Collins did a study. There's one 3 days, I think 7 days, 2 weeks, and then she had a 12-week and a 28-week as well. And so the results kind of seen at the 12 and 28-week were pretty similar. And so at that point, it seems like obesity has been induced. And then the first kind of time points is just more of like a introduction to obesity.
Thanks for the presentation, Hannah. I just had a question about the fiber, like the delayed fiber intervention, about how does that like 3-week time period translate to humans?
Yeah, and so I don't know how translatable it is to humans, but at the 3 weeks, obesity has already been induced, and the animals already experiencing the effects of obesity. As I mentioned, we have done studies in the past that look at the effects of obesity in the shorter time frames, and so by 3 weeks, we have seen obesity be induced, and they're significantly bigger than the chow control animals, and so I don't know how exactly to put that in the same time frame as humans, but it does show. It does, the kind of point of it was we wanted obesity to already be induced by the time we started the fiber intervention. Thanks.
Just to kind of follow up on that, is there any studies like in humans showing that, say if someone just totally changes their diet and then has really high fat, high sucrose like diet, how long it takes to achieve obesity?
I'm not sure. No, I don't know about that. Sorry.
Other questions? Yeah.
More basic question. Why is the slow twitch soleus muscle more resistant than the fastest muscle to fat infiltration? Yeah, I'm not exactly sure what the mechanism is. I think it has something to do with it like using the fat better, because the slow twitch, they use fat for energy better than the fast twitch muscles, but I'm not sure what the exact mechanism is.
Do sarcomeres turn over faster in non-oxidative muscle than oxidative muscle?
I'm not sure.
It just kind of like renews itself faster.
I'm not sure about that.
All right. Deep thought. You speculated that the estrogen might steer the fat infiltration away from visceral. Yeah, away from visceral. Could you confirm that? Like, I would assume that you could just go in there and start looking at visceral fat between males and females.
Yeah. We probably could. We definitely have to like normalize for like total body mass, and that's something we couldn't now do retroactively. I think we would have had to consider that in the experiment at the time. But definitely possible because we do collect fat, so we could just weigh it, and we do weigh it, but we don't collect all the fat. We just collect some and we put it into tubes and we weigh it. But yeah, so we would have to harvest all of it, but it would be interesting to do that.
I got one other question. I'm sure you don't have the answer, but it's something that I thought about. So, you know, the human body seems like pretty smart. It does things. So one of the reasons that, you know, we start storing fat is so we maintain energy.
Yeah.
So it just seems like fat has its benefits.
Yeah.
What is the benefit of fat releasing inflammatory cytokines?
I'm not sure. I'm sure it has, or it had some purpose.
Yeah.
At some point, but yeah, I don't know.
I was just gonna ask if, like, the fat does secrete inflammatory cytokines, but it's also the macrophages with the fat as well. So it isn't necessarily the adipose tissue, like the adipocytes. And in terms of the benefit, there's a lot of research looking at more like day by day, like day-to-day changes in diet. So it is really helpful to store a lot in terms of like evolutionary perspective of like, you're not going to eat for a little bit. So you're able to increase more adipocytes, like have adipogenesis, so more adipocytes, and then hypertrophy as well. But then you also want to be able to bring that down. Yeah. So the dynamic part is important.
Okay. So could it be that the macrophages are that sort of the beneficial way of keeping that in check?
Yeah. Yeah, exactly. Going back and forth.
Nice.
To add to that, I would have a similar idea. I mean, I don't really know what the answer is, but fat is very expensive because it has a very heavy blood supply. And so when you have a lot of fat, a lot of the blood supply that you have will be diverted away from other organs. So at one point, presumably, you know, having a little bit of fat is good. In fact, we know that we need some fat to survive, but I guess that might be a natural response. When you have too much, then it gets very costly, and the body wants to keep that in check somehow.
Other questions?
Oh? Well, we've got an online question for you, Hannah. If the 3 weeks fiber delay group is given 12 weeks fiber supplementation instead of 9 weeks fiber supplementation, would you expect some different result?
I don't think so. I would expect the same result. I think because they're still having the high fat, high sucrose diet, I think it's having some positive effects on the metabolic syndrome, but I think they're still going to be gaining fat, and the fat seems to be, or the increase in body fat seems to be what is increasing intramuscular fat. And so I think with an extra 3 weeks, they're still going to be gaining fat. And so I don't think that's going to mitigate the effect of the high fat, high sucrose diet, just having the fiber for another 3 weeks.
Do you think that like age would have an effect, like not necessarily on the sex effects, because like menopause and things like that would have an effect, but if you like 12 weeks is quite young for rats, but if you did this in like way older rats, do you think that, what do you think the effect would be? Yeah, and so there's an age effect as well with the intramuscular fat infiltration, as like intramuscular fat infiltration or sarcopenia is even typically seen in like the older population in general. So there would definitely be an age effect, and I think kind of as you age, I think even the control animals would start to experience an increase in intramuscular. I don't think it would get to the same point as the animals on the high fat, high sucrose diet, but I think they would start to have an increased amount as well.
I think Nada's doing that study. She has some older animals. I don't know if anyone's looking at the muscles for them though, but I know she has some older animals that she's going to look at.
I may have missed this, but so you mentioned that there was a widespread in how the different rats reacted, and maybe that may have influenced your statistical result. Did you normalize for anything to kind of help with that, or was that not possible?
For the way we calculate the intramuscular fat percentage, it is normalized to like cross-sectional area of the muscle. So it's a percentage of the area of the muscle. So it's already kind of normalized to the size of the muscle, which is, I guess, representative of the size of the animal. So it's just the way we measure it is, I don't think it needs that like extra step to be normalized. Yeah. Dana?
Thanks for the presentation, Anna. Did you look at feeding behaviors at all?
Yeah, we did look at feeding behaviors, but I'm sure, as you know, it's sometimes very hard to look at them because they flip their dishes over and they are always eating everything. And so it's not necessarily like representative of what they're eating. And so we do have that data, and I haven't looked at it closely. I know Mauricio's looked at it a lot closer for his animals. But for me, like, I didn't spend a lot of time looking at it because I didn't think it was going to be super useful because of how, like, there's always just spilled food everywhere. And I just didn't think the data was going to be very meaningful if I took a look at it.
Totally. And then also, did you do anything with the liver?
No, we didn't do anything with the liver.
Did you notice a lighter color?
Yeah, definitely. And yeah, on some of them, I know like maybe like Ruth could speak to this a little more because she looked at the liver a little closer when we were harvesting. But I know something we'd like look for in the high fat animals was like the spotty liver, and if it was lighter in color.
Okay, I was just really interested in the prebiotic delay
Okay. Yeah.
What that might have done too.
Yeah, I'm not sure. I definitely I didn't take note of maybe a difference in the liver between those two groups, but I'm sure we have the liver. So yeah, we definitely harvested it and have it. So
about 2 or 3 summers ago, a summer student looked at the liver, and in the high fat sucrose animals, you see big fat deposits in the liver compared to the cow animals, but it was not really of a primary interest for us. So we know that happens. What kind of implications that might have, for example, for the metabolism and other things, which might integrate because the liver is so important. Yeah. But you never really check that. But there's clear differences.
Or this might be kind of a naive question, but you used prebiotic fiber supplements.
Yes.
If people use probiotic supplements, is that an option, and what is the difference?
Yeah, I'm not sure. I haven't looked into like probiotic supplements. Probiotic supplements, from my understanding, are actually like introducing bacteria into the gut, whereas this is just feeding the gut bacteria with like beneficial fibers that increase the growth of like good bacteria in the gut. And so I would assume it would have either similar or maybe not as good of results, because even if it's not being fed, I don't know, Dana probably knows more about this, but if it's not being fed with like the beneficial ingredients to like increase the like good bacteria, I don't know how much they would like sustain, like if they were given a probiotic, if they would like sustain like high levels of that bacteria.
It's really interesting because we do that because it's kind of like a backward thing, because Raylene has done those type of prebiotic fiber interventions with humans, and then we tested it out in animals to potentially look at some mechanism. Normally it's the other way around. You do first animal work and then you go to humans. And here we took something over that Raylene had found to be fairly successful in a variety of ways in obese people.
And that's what I was wondering too, because I think there's also work done on like a combination of the two, like a symbiotic, where there's a pre and probiotic. I was wondering if that was the case, would the delayed effect maybe be less problematic, or maybe having both in there helps even if you're too busy? I don't know.
It's possible for sure.
Dana.
Yeah, I think what Hannah was saying with the probiotic, like it would have to be chronic, and it's likely that the probiotic, like it would be like a supplement every day, which is the same as the prebiotic, and it's likely that the probiotics would just be like replenishing themselves. They're not actually establishing a colony of themselves within the gut, and so what usually happens is once you stop taking those probiotics and you're not eating the probiotics, like the actual bacteria in your gut, the substrates that it needs, which is like the prebiotics, then they are going to die off, more or less.
I know you did not look at the mechanical properties, but do you believe that the functional properties of these classes are we talked about chronic obesity at some point, do you think there is an effect on the mechanical properties?
Yeah, I don't know if I can say based off of like my animals, I don't know if I saw any difference in like functional movements between the high fat, high sucrose group and the chow group. But I do know in the literature they've seen even like they've seen over and over again that the obesity does lead to a worsening of physical function. And it's even been shown like on the level of like single contractile properties that if adding like fat to that environment will like decrease this like contractile properties. So I think yes, but in terms of like physical limitations, they were behaving very similarly. And so I'm not sure if in my animals there was any like real like physical limitations to the group with the high fat, high sucrose diet.
Just to kind of follow up on that, did you measure, I might have missed it, but did you measure the mass of the muscles?
Yeah, we measured the mass of the muscles. Again, I didn't look too in depth into that data. I think I looked at it one time, and just after having experience dissecting, they can be very, how you take them out of the animal can vary from person to person. And there was a couple of different people who'd done the dissections for these animals. And so I wasn't confident that I could say that they were consistent so I could measure them against each other. So I didn't look at that, but I could.
That would be interesting because I think there's in humans, the excess body weight like increases the strength requirements, and then there's an increase in cross-sectional area of the muscle, and then that could potentially compensate for that muscle fat infiltration. So it'd be interesting to see if that like also occurred in humans.
So Stephanie, I mean, you know, with all the difficulties of being very consistent in taking the muscles out, but just kind of on average, what we seem to be finding for when we have measured that is that in the fat animals, they have bigger muscles. And then when you normalize it to body weight, then it seems to be about the same. That's what we seem to be finding. But there are limitations. Those are very small muscles. You know, take a little bit more of the muscle, a little bit less. You know, that makes a 5 or 10% difference immediately. So it's a bit tricky to be very consistent.
Hannah, did your animals have access to a running wheel?
No, they were just housed in cages, and they weren't getting any exercise other than just kind of running around. But cages were small, so not anything crazy.
And you mentioned like not really noticing any just like observation or like any behavioral.
No behavioral differences.
Do you think, I'm speculating, like do you think like if all of them had access to a running wheel, like do you think that would have affected your general results? Yeah, the access to a running wheel is interesting because there has been we have done studies where they're like made to run on a treadmill, and so if access to a running wheel, I don't know how motivated they would be to run on the running wheel, but I do think if they were exercising, it would have quite a difference on the results like a positive, like they would decrease the fat.
Yeah. Do you think that would interact with some of the diet interventions where you see like a small maybe improvement based on like the prebiotic that maybe that's not doing enough on its own, but in combination with them maybe being more active or being able to stay? Because I know I've seen some studies where like rats that end up obese like stop running. Like, do you think maybe there'd be like a behavioral interaction there that could help?
I'm not sure.
So when we had rats that received a high-fat sucrose diet, when we had them run from the very beginning, when we exposed them to the high-fat sucrose diet with similar protocol, they would be running, and interestingly, the muscles and the joints that we looked at, they seem to be good. So the musculoskeletal degeneration seemed to be gone in the presence of a high-fat sucrose diet when they ran. But interestingly, they still became big. They were not quite, they didn't have quite the same body fat percentage, but they clearly had a higher body fat percentage than the animals that had a regular, rat diet. So they were running and they got the bad diet, the high-fat sucrose. They gained weight, they gained body fat, but the musculoskeletal system seemed to be preserved. And the big difference was when you then looked at the metabolic profile, that seemed to be all normalized in the running rats, which suggested to us at one point, it doesn't really matter so much, how much fat you have, but whether or not that's associated with metabolic syndrome or not, that seemed to be the crucial thing.
Yeah. I just wanted to ask if there was also infiltration of fat accumulation in the muscles in those studies?
No, that was,
That's very, just because whenever like seeing studies with rodents, like I know I've done like no rodent work myself, but it's my impression that they really like to run a lot. So it seems like the natural state of the rat is to be running. So like, I guess, and it's good for your model to have all of them be sedentary. It's like on top of being sedentary, but it's always interesting to see like the sedentary part is already kind of a treatment in and of itself. Kind of.
And you are right, I've said that several times. Our dream still is to get these metabolic cages for rats where they can have access voluntarily to the wheel, or we can force them to go in there and we can run it on a motor and we can measure oxygen uptake all continuously. Unfortunately, they come with a certain price tag that we haven't been able to afford it. But I mean, at the end of the day, that's what needs to be done. You're absolutely right. When you read reports of free wheel running, rats seem to be doing that 2, 3, 4 hours every night. So they run much more than the half hour or hour that we force them to run. So, and it would be interesting to see. And as I said before, when we expose them to high-fat sucrose diet, we have them run. It's not a problem. But some of the animals that we had up to 28 weeks in a high-fat sucrose diet, they wouldn't be able to run anymore because they got so big that they supported part of their body weight with their belly, and they used the leg to kind of propel them forward like in a rowing, So they were not really walking anymore. They kind of propelled themselves forward on their belly, and they couldn't run anymore once they are somewhere really big.
More questions?
I have the last question. Did you look at the lean mass, like the percent?
Yeah.
With the body fat?
Yeah.
Okay.
It was very, it was correlated to the inverse of the body fat percentage.
Okay.
It all lined up pretty well. But I have it. If you're interested in looking at any of the data, I have it.
Yeah. And you guys did gut microbiota, right?
We did, yes. I think Nada's taking a closer look at it right now. Because we did look at the gut microbiota.
You looked at differences in tendon, or like even the previous studies like Kelsey or anybody looked at tendon differences?
I know there has been a couple of studies looking at like the tendon strength. I don't know what the results are off the top of my head, but I know it has been done.
Any other questions?
So your results seem to suggest that something quite dramatic happens in the first 3 weeks there, and so do you think all is lost, or do you have a suggestion what might be done? Because you become obese, you have metabolic syndrome, then you want to do something about that. And are you kind of saying that it doesn't really help whatever you do, or?
I think it would be interesting to go the other way and try the exercise intervention and delayed exercise intervention, because I do think those are like different pathways, whereas this fiber is very like gut microbiota and metabolic, whereas the exercise has a lot of other benefits, and as well as like, yeah.
So you're saying that the delayed exercise intervention?
Yeah, a delayed, I think it would be more successful than a delayed fiber. But yeah, I don't know for sure. The results that I got do suggest that it might not be rescuable. So yeah.
What do you think? You've never done that, and maybe what would happen if we do the 12 weeks of high fat sucrose and they get really big, and then you just go back on a regular diet?
Yeah.
So you don't do anything else. You just have a bad diet for a while, and then you get back to a good diet.
I think that would be interesting. I think that would have more of an effect than just implementing the fiber, because they're still on the high fat, high sucrose diet. And so I think maybe going back to a regular diet would be more beneficial than just implementing a fiber intervention. But I'm not sure.
That's a really good question. I was just about to ask that because we don't see a lot of people doing that. Like, as the intervention of like just going back to their regular diet. We were actually having this discussion in our journal club this week. Because I think it would have a really large effect, because the chow has fiber as well, a lot more fiber than even the regular like processed low fat diet. But then it also has that decreased caloric amount, and then also like changes in fat, etc. So I think it would make a really big difference, but it's possible that there are some like irreversible kind of like propensity inducing effects of like then going back onto the high fat, high sucrose diet. They might have an increase in obesity further than someone who only had like one consult and continued for the rest of their life onto a control diet, control for their life. But
There are studies on that, isn't it? Like cyclic dieting, where you like keep accumulating more than if you just stayed on a high worse diet.
Yeah, we don't have a lot of supply. Like look specifically at like fatness tissue or muscle within that.
Any last questions?
Just a very small question. You mentioned GLP-2 and LPS markers, and we have the blood, don't we?
We do, yeah. Yes, we do have blood. We've analyzed a lot of it, so I don't know how much is left. And then I've also heard, I've talked to Raylene about it. LPS is the assay for it's very finicky. So you need a lot of sample to kind of redo to make sure you get it right. But we do have some sample left, so it is possible. But again, it's just a 3-week time point. We don't have it before 3 weeks, because it would be interesting to see kind of, like if we took it like 5 days and 10 days and 14 days. To see how it changes.
You have a resting level. I mean, you have a 0 level.
We have a 0, yeah.
All right. Well, let's thank Hannah again.